Okay, so lecture part 2 of chapter 16, which remember are non-specific, or what we call innate defenses. Okay, so innate immunity. So last time when we left off, and fingers crossed that my dogs aren't going to bark, we'll see what happens. We're starting to talk about blood, okay, and that the formed elements were made up of white blood cells, red blood cells, and platelets. These are all coming from your bone marrow. And then plasma is pretty much everything else that travels around in your blood, whether it's vitamins, electrolytes, etc. Okay, so now we're going to talk about the different types of white blood cells. So these are leukocytes. So we were right here where we left off. So I had mentioned these terms before, but I want to reiterate them. So remember, when you're looking at a lab report that you get back from a patient, the typical number of white blood cells that are found in a cc or milliliter of blood are anywhere between 4,000 to 10,000. So if you see white blood cells that are above the 10,000 mark, that indicates leukocytosis, and that means that your patient is fighting some type of infection, whether it's bacterial, viral, whatever it may be. So if you have a patient and their, lab report comes back and they have, 18,000 or they have, 25,000, that indicates an infection. So leukocytosis, increased number of total white blood cells. Leukopenia is the opposite. So this would be, maybe the patient now has 1,000 white blood cells. So that can indicate active HIV infection. It can indicate aplastic anemia, which is basically where your bone marrow goes on strike and doesn't produce anything. It can indicate some type of genetic disease that is affecting their white blood cell production. Something like that. But these two are opposite. I think this is what you're most commonly going to see in your healthcare careers. So knowing the total number of white blood cells is awesome. But then if you take it a step further and do what is called a differential white blood cell count, that can give you even more information. So this is basically looking at 100 white blood cells and then dividing them up into the different categories, okay? And there are 5 categories of white blood cells. And thankfully, this is automated, but clinical lab scientists are also taught to do this by hand by looking at a blood smear and just looking through a microscope. So if the machines go down, the scientists can do it as well. So there are 2 main categories, and so you might want to label these 1 and 2. The first big category is the granulocytes, okay? So maybe label that number 1. And then category number 2, which is a couple slides ahead, are referred to as agranulocytes, okay? So the only difference in the spelling is there's an A as opposed to not. And all this means is that when we look at these cells under the microscope, they have like chunks of stuff in them. They have granules, okay? And the agranulocytes do not have granules, okay? And we're not gonna go into what the granules actually are. It's just a way to tell them apart. Also, I will never ask you anything on a quiz about, what color are the granules in neutrophils? We don't need to worry about that, okay? That's nitpicky. So just know that they fit into that category. So the definition of granulocytes are white blood cells with large granules when they're looked at microscopically. And there are 3 categories of granulocytes. Okay, so you might want to label these A, B, and C, or 1, 2, 3, whatever is easiest for you. Number 1 are the neutrophils. Number 2 are eosinophils. Number 3 are basophils. The ones that we are going to talk about the most and which are most predominant in your immune system are the neutrophils. Okay, and another term that you're going to hear me talk about a lot is the term phagocytosis, which we touched on briefly in chapter 6, but Phagocytosis is the process of cells engulfing material. So you can kind of think of it as cells eating. And neutrophils are highly phagocytic cells, meaning that when they see clumps of things, and especially things that don't belong and are not supposed to be in your system, like clumps of bacteria or clumps of, viruses or damaged cells, they are going to engulf and they're going to eat that stuff, which is great for you because it means it's clearing it out of your system. Okay, so neutrophils become highly phagocytic and they are really important in fighting infection. Okay, eosinophils are, what they do is they don't engulf anything. They can a little bit, but they're not highly phagocytic. But what they do is they secrete toxic proteins against parasitic worm infections. So whether it's a big old tapeworm or, a microscopic hookworm, remember worms are eukaryotes, and so they're still really big in terms of looking at things under the microscope. And so there's no way that there's gonna be a cell big enough that can engulf an entire worm, even if it's a microscopic hookworm. So the way that these cells work is they produce these toxic proteins that kill and basically poke holes in the worm. And then as the worm degrades, then they can phagocytize some of those smaller pieces. Okay, so if you have a patient that comes back and they have a highly increased level of eosinophils, what that indicates to you is they could be battling a parasitic worm infection. Now, you can see increases of eosinophils too in inflammatory conditions, whether it's an autoimmune disease, but it's usually in correlation with other types of markers that are in the blood. And I'm just talking about the main, what these cells basically do. So for the scope of this class, if I were to ask you a question about what does an increased eosinophil count tell you, the answer would be that, the patient possibly has a parasitic worm infection. Basophils are they're probably the ones that are in the lowest concentration, and what they do is produce histamine, which remember is part, and it's absolutely essential to a normal immune response, and especially triggering inflammation. So remember, histamine is only bad when it's way, way, way overproduced, such as what happens in allergy. But you would not want to be histamine free because your immune system would not work the way it's supposed to. So this is kind of what they look like. So if we look at different types of cells, okay, here's your red blood cells. They don't have a nucleus. Remember, they are only there for gas exchange. When we look at the granulocytes, okay, and you can see these are the big chunks of granules that are in here, and trained med techs or clinical lab scientists are able to tell them apart. I didn't know if I told you guys this, but I actually, when I started out in college, I thought I wanted to be, a clinical laboratory scientist. And one of the things that kind of weeded me out was I could not tell the difference between these under the microscope. And that's kind of a big deal. So that kind of told me that fate was, steering me in a different direction, that I would not be a good med tech. So anyway, so the majority of your granulocytes and really white blood cells are going to be neutrophils. And the whole purpose of neutrophils is phagocytosis. So what these guys are going to do is when they get a signal, they are going to go to the site of the infection. Okay, let's say, for example, you get a puncture wound in your arm. Some Staph aureus got in with the puncture wound, and now you've got Staph aureus growing and you've got an infection started. Once the neutrophils get the signal, uh-oh, we have a problem, they are going to travel to the site of that puncture wound and they are going to start eating stuff that doesn't belong, okay? That is their primary purpose. Basophils, the lowest volume, these are the guys that produce histamine when you need it. And then eosinophils, these are the ones that increase when there is a parasitic worm infection. All right, so now let's go to the agranulocytes. And these are the guys that don't have any granules. I think they still look like they have granules, but that's why I'm not a med tech, okay? So there are two main categories of agranulocytes. The monocytes, which we are going to talk about in this chapter, and then lymphocytes are the ones that are involved in your specific immune response. And so these guys, we're going to hold off and talk about them in chapter 17. Okay, so lymphocytes, which you'll also hear them referred to as B cells and T cells, that's chapter 17. They do not eat anything. They're not phagocytic. They are the ones that are going to produce antibodies and, are involved in your specific immunity. Okay. However, really important in this chapter that you understand what monocytes do. Okay, so monocytes transform into huge, gigantic cells called macrophages. So macro means big, phage means that we're going to eat stuff. So the neutrophils are very phagocytic. These guys are way, way, way more phagocytic. They can eat a lot of stuff, and they are huge in size compared to the neutrophils. Okay, here's the difference though. So if you've ever, heard the story of Superman, so Superman goes through life as Clark Kent, right? Kind of a nerdy reporter. And then when he needs to transform into Superman, he steps into a phone booth, which FYI were little booths that had phones in them. I know you guys, being as young as you are, probably don't even know what those are, but the point is he had to go through a transformation from nerdy Clark Kent into Superman. And that is basically what happens with monocytes. So when monocytes get activated, and normally it's when they detect things like histamine in the bloodstream, they take time, but they transform into these giant cells called macrophages. And then the macrophages head over to the site of the infection, the crime scene, as I like to call it, okay, where that puncture wound is. So the neutrophils go there first, followed by the macrophages, and it's just because macrophages take time to transform from monocytes into macrophages. So here's what the agranulocytes look like. So I guess there's no granules in here. To me, they look exactly the same. But here are the monocytes, and these are the ones that are going to become huge and turn into macrophages. And then your specific immune system, lymphocytes, and we'll get to these in chapter 17. And then these are what platelets look like, okay? So they're not really cells, they're little hunks of cells, absolutely essential for blood clotting. So we have two types of phagocytic cells, okay? We have neutrophils, and then we have monocytes, which are gonna turn into macrophages. Now, both of them are gonna head to the site of the infection. So the neutrophils, because they don't need to go through a transformation change, they're gonna get there first, and they're gonna start eating stuff that looks foreign, okay? Whether it's bacteria, viruses, fungal cells, whatever. The monocytes, in the meantime, are gonna be changing into macrophages, and then they will arrive a little later at the site of the infection. Now, luckily, and this helps your non-specific immune response as well, you also have macrophages that are fixed in your body in certain tissues and organs, most notably your lymph nodes, okay? So when you're fighting an infection, you'll notice that, your lymph nodes get swollen, they get kind of tender, and that's because the macrophages in those tissues are starting to filter, whatever's causing the infection out. And so, actually, when you have tender lymph nodes, don't look at that as a bad thing. Look at that as, yay, my immune system is working and my macrophages are doing what they're supposed to do, okay? It doesn't mean that it's pleasant. I mean, it's not fun to have tender lymph nodes, but it means that your immune system is working. You also have macrophages that basically wander around your bloodstream and they look for anything that looks foreign, and they're going to get it out of your system, hopefully before it ever causes an infection. Okay? But primarily, the macrophages, once they're transformed, are then going to head to wherever the infection is. Okay? So the puncture wound that I'm using as an example. So these macrophages, so here's a photo of them, and I think I've shown you this earlier when we were talking about the difference in size between eukaryotes and prokaryotes, but macrophages are gigantic. And so here's the little bacteria. I mean, you almost feel sorry for it, but the macrophages have these pseudopods that, you know, are not these big, thick, lumbering amoeba-like things like you see in textbook, but they are like, you know, little arms that come out and they grab that bacteria with a stranglehold and they pull it inside the cell, and then they're going to digest it basically it's going to fuse with something called a lysosome, and that's going to have enzymes that will break down that microbe. Okay, so one macrophage can eat a lot of bacteria or whatever the microbe is. So how does this process occur? How do neutrophils and macrophages know to get to the site of the infection and to start eating things? Well, the process is called chemotaxis, and we had talked about this also, this term in chapter 4. So if we break this down, chemo just means chemical. Taxis means movement, and I remember that because I think of a taxi. You know, you get in a taxi to actually go somewhere. So there are going to be certain chemicals released into the bloodstream that are going to alert, first of all, the neutrophils, hey, we got a problem, you need to get over here. And then also the monocytes to start their transformation into macrophages. Now, what are some of the chemicals? We refer to the chemicals that start this process as cytokines. And by far, the primary cytokine is histamine. Okay, so histamine, you know, your cells wouldn't know to get to the site of the infection if you did not produce histamine. So once histamine starts traveling through the bloodstream, which will happen when your defenses have been basically bridged by a microbe, that histamine is going to trigger the neutrophils to get to the site of the infection, and it's going to trigger monocytes to start turning into macrophages. Now, there are other things that can do this as well, other types of cytokines. So basically anything produced by the microbe. So let's say it's a bacteria that produces a toxin. That toxin traveling through the bloodstream is also going to function to activate the neutrophils and the monocytes. Also, maybe parts of the cell wall that have been damaged, or it can be anything in your cells that should not be floating around by itself. Okay, so if there are free ribosomes floating around in your bloodstream, or mitochondria or other components of your cell, that indicates that something is wrong, because that shows that your cells are being damaged and they're starting to leak. So those things can also function as cytokines. And all of these together are going to turn on the early warning system to the neutrophils and to the monocytes that, you know, we got a problem. You guys need to get to the site of the infection and start doing what you're supposed to be doing. So then what's going to happen is the phagocyte is going to feel around, and it's going to determine that, okay, this is actually a foreign invader. It's not part of me, okay? And that process is called adherence. Now, remember when I talked about the function of envelopes on viruses or bacteria, and also capsules? This is where the pathogen is able to evade the phagocytic cell, because that envelope and that capsule is going to feel a lot like one of your cells. So eventually the neutrophils and macrophages are going to figure it out, but this buys the pathogen a little bit of time. So, you know, that's why capsules and envelopes are known as virulence factors, because they're going to interfere with the phagocyte a little bit. One of the things that really, really helps, and we're going to talk a lot about antibodies in chapter 17, but any cell in your body that has an antibody on it, that is a huge signal to a phagocytic cell, and it basically is waving a big flag that says, hey, I have an antibody sticking to me, I am bad, you need to eat this and get it out of my system. And that's why vaccination is so, you know, works really well, because when you're vaccinated for something, you're going to have these circulating antibodies that are going to glom on to the surface of the pathogen. And so hopefully the phagocytic cells are going to eat it before it ever infects you or causes any damage. So keep that in mind too, but we'll get to much more of a discussion of antibodies in chapter 17. Then what's going to happen is those little arms, or the pseudopods of the phagocyte, are going to pull the microbe inside, and they're going to enclose them in a vessel called a phagosome. So it's just kind of a little bubble inside the cell where the bacteria or the virus is enclosed, and then it's going to fuse with an organelle called a lysosome. Okay, and the lysosome you can think of as basically being kind of a cellular stomach. It's just a little compartment in the cell that is full of digestive enzymes and maybe hydrogen peroxide. And so when the phagosome fuses with the lysosome, those digestive enzymes are going to go to work and they're going to break down that microbe. So normally they will kill or inactivate, you know, bacteria, virus, fungi in about 10 to 30 minutes. And then the cell basically kind of throws up and, you know, barfs out the parts, and then it can go on to its next victim, you know, and try to clear more things from the infection. One thing that I want to mention though, okay, so sometimes macrophages don't barf out everything. Sometimes macrophages will take parts of microbes that they've digested and display them on their cellular surface. Okay, kind of like you probably, you know, if you've ever watched a crime show about serial killers, one thing that serial killers do is a lot of times they will take trophies from their victim, you know, like, oh, I have to keep this earring, or I want to keep her driver's license. It's kind of the same thing. So some macrophages keep parts of the microbes they eat and they display them on their surface like trophies. I want you to remember that because it's going to come into play when we get to chapter 17. Okay, so here's the whole process. So we have our microbe. Eventually, the phagocytic cell is going to figure out, hey, this is bad. It's going to bring it in and enclose it in a little vesicle called a phagosome. That is going to fuse with a lysosome, which is full of digestive enzymes and hydrogen peroxide. And those two fuse, they digest everything, and then the phagocytic cell poops it out, except for macrophages, which sometimes like to take these parts and display them on their cell surface, which I will explain in chapter 17. Okay, so this is another example of how huge the phagocytic cell is. Here's the little bacteria. So one phagocytic cell, and especially macrophages, can eat a lot of stuff. It's not like they're going to eat just one little bacterial cell or one clump of viruses. Okay, so I'm going to stop there because we're at 24 minutes and get this uploaded. We will pick up with extracellular killing, which refers to eosinophils, in part 3. Thank you.